welcome to Science Side Up. Today we're going to stir the gene pool and serve up sizzling science. I'm EJ.
And I am Jerwin, your co host and fellow science nerd. Today, we're gonna talk about pigs. Not just any pigs, genetically inbred pigs with secrets hidden in their DNA.
Exactly, sir. Welcome to our topic
Roommates for life, pig genetics, and inbreeding tales.
Yup, we're diving into the ROH or the runs-of-homozygosity. It sounds fancy, right? But I want you to think of it as what happens when the big chunks of DNA start repeating themselves. Like, the deja vu in your genome.
Right. Copies of the DNA say the exact same thing. No remix, no variation, just a straight repeat across the chromosome.
Agree with that, sir. And the main culprit?
Inbreeding. Inbreeding happens when closely related individuals mate. Think like brothers or sisters, cousins, the family ties that are way too tight.
Exactly, sir. And when that happens, their offspring don't get a fresh mix of the DNA. Instead, they inherit the same version of the genes, from both
Yeah.
sides of the family.
That's right. And here's the catch. The more this repeats generation after generation, the more the genome starts to look like a photocopy of itself.
And when genetic variation goes down, it means that the homozygosity or the identical gene copies, it goes up, right?
Right, yes. Which bring us right back to ROH. Those long, identical stretches of DNA we talk about are basically the genetic footprints in inbreeding.
And since we talk about the ROH, we have, we got an interesting study here. It's based on the Tao et al. BMC Genomics of 2025. The title is The Effects of Runs of Homozygosity on Pig Domestication and Breeding.
So let's start with the methodology. The methods in this paper are pretty technical. So let's break them down into simple, bite-sized chunks we can all absorb.
Okay, first collecting pig DNA and traits. First, the what they did, I mean, is they collected the DNA from more than 7,200 pigs. Okay, that includes breeds, domestic breeds, wild boars, and even feral pigs from 41 countries. It's a very big data, kung makikita natin. So, they use special DNA chips, like Illumina Porcine SNP60 and the GeneSeq GGP chips to scan the genome. Those are scanners. Missing genetic data, kapag meron naman silang na-miss na data dun sa 61,114 autosomal genomes na binabasa nila, okay, no problem, because they are going to use a reference collection, okay, that is what they call the Shape It 5 to fill the gaps. Okay, so on the top of that, they recorded those traits like body size, reproduction stats, meat quality, and even risk like scrotal hernia in order to understand if there's a connection, if the ROH affect the gene expression, the traits, and so on. So, on your part, sir.
Next, they turned thousands of genetic markers into a few easy-to-see patterns using the PCA, the Principal Component Analysis. They used the tools Plink to crunch the numbers and PCA tools in R to make visuals. The results? They confirmed that pigs were domesticated independently in Asia and Europe. They also spotted inbred lines like the tightly clustered ZXV pig breed.
Now let's talk about this one, detecting ROH or runs of homozygosity. That's when a pig inherit identical DNA stretches from both parents. It's a clear sign of inbreeding. They scan genomes with plain, check for related samples using the king, and use for detect runs to measure how often ROH appeared. Now, why does it matter? because the ROH regions might carry a harmful mutation or influence important traits.
After that, scientists use the tools like the GMMAT and the GMMA to link genetics to traits, and VCF tools to check genetic differentiation. Sure enough, more ROH was tied to smaller body size, narrower hips, and higher risk of conditions like scrotal hernia.
Studying the gene activity, but it doesn't stop at the traits. ROH also affects how genes turn on and turn off. They analyze gene expression in pig liver, in muscle, in intestine using the RNA sequence data tool. Okay, tools that are involved are Trimomatic, HESA2, StrengthTie, GAT-K4, they all handle the data, while the cluster profiler check which pathways were involved. So the findings, ROH influenced thousands of genes, especially those related to immunity and the development.
To wrap it up, the researchers collected DNA and trait data from thousands of pigs worldwide, scanned their genomes with DNA chips, used computer tools to fill in gaps, mapped genetic relationships with PCA, and detected inbreeding through ROH, and finally linked those patterns to traits and gene activity. Now, to better understand what all these methods revealed, let's move on to the findings.
Did you know that pigs were domesticated separately in Asia and Europe? That's why if you're going to look at their ROH patterns, they are so different.
Oh yeah, European pigs are like, oh, we've got diversity, while the Asian pigs are more like, oh, we've got ZXY pig, 20 generations of inbreeding.
Exactly.
Yeah.
Now, I want you to look at the figure 2, sir. So each point here in the pig breed or populations, x-axis and y-axis are first two principal components from what they call the PCA. That's just a fancy way of capturing the differences or the biggest differences in the DNA.
Oh yeah, see, let's see.
Okay.
See how the Asian pigs cluster on one side and the European pigs on the other.
I see.
That clear split shows they were domesticated independently.
And now notice how the Asian wild boars are spread out more widely.
Okay.
Look on the figure. That means it has a greater genetic diversity. And meanwhile, in the European pigs are packed tightly together. If you're going to look at
okay.
the figure, which suggests it has a stronger artificial selection. And yes, a more inbreeding.
Yeah, and see, those points in between, that's where feral or wild populations land. Genomes that are kind of a mix, not fully selected like the domestic ones.
So this figure isn't just a pretty plot, right?
Yeah.
So it's a map, or it's a map of the pig history, their DNA. It shows how the domestication and the breeding shape their genetic diversity.
Yeah, that's right. Which bring us back again to ROH. The tighter those clusters, the more we can expect a copy-paste DNA showing up in the genome.
Exactly. So
Yes.
I agree with what we had or what we talk about on this part. Now I want to talk about these inbreeding. Okay? So ROH isn't just some curiosity. It is a link to the real biological problems in pigs.
Right.
We're talking about the scrotal hernia, we're talking about the low birth weight, even higher piglet mortality.
That's right. So here, basically, the more ROH, the more trouble. It's like your genome saying, oh, I'm tired. Please stop marrying your cousin.
And
Yeah.
please stop marrying your cousin.
Yeah.
Exactly. Now, here's where the science gets sharp, or it gets more clear.
Yeah.
I want
See?
you to look, sir, on the figure 4.
Okay.
Now it shows how the ROH burden measured as the fraction of runs of homozygosity, or what they call the FROH, or the portion of the genome inside the ROH. It affects the traits that really matters to the breeders, to the farmers.
Oh yeah, that's right. Each bar here represents a trait, like what the breeder wants to show in their pigs, like the growth traits like body weight and length, reproductive traits like birth weight and piglet survival, and even disease risk, like the scrotal hernia and the post-weaning diarrhea.
And
Yes.
for example, in Eurasian pigs, the higher the ROH, it strongly suggests or strongly increase the risk of the what they call scrotal hernia. So meanwhile, in Sodian pigs, more ROH means that, or meant, or it could result to a what we call smaller hips, of course a narrower chest, and shorter bodies and lighter weights.
That's right. And it's not just about the size. The reproductive traits are hit too. Average birth weight drops, along with the piglet mortality shoots up. The statistics back it that 6 associations were significant, way more than expected by chance.
Exactly. And so what we talk about here in this paper
Yeah.
is a message, or it's a clear message about inbreeding depression. It says that
Yeah,
it's
that's-
real.
Indeed.
It is real. And ROH doesn't just a mark of the inbreeding. It exposes the hidden recessive mutations in the genomes. It is affecting, or it's dragging down the growth, the reproduction, and the health.
That's right. In pigs, in other words, the ROH isn't just a genetic diary anymore. It's a warning label.
It's a warning label.
Yeah, warning.
Exactly. It is a warning label. It gives us information
That's right.
about the inbreeding history. Now, on this part, sir, let's talk about how the ROH, based from the paper, how the ROH rewires the genome.
Okay, let's see.
Okay, let's talk about this one. So far, we've seen how the ROH shapes traits and health. I want you to look now in the figure 5.
Okay.
Okay, it takes us to a deeper understanding. It shows how the ROH actually rewires gene activity across tissues.
Okay, that's right. Yeah, let's see on the panel A. So first, the inbreeding levels. Here, we can see the 3 pig breeds. We have the Duroc,
Duro, yeah.
the Landrace, and the large white.
Mm-hmm.
This says that some pigs have way more ROH, meaning they are more inbred.
And I want you to look at this one on the panel B.
Yeah.
And that extra ROH doesn't stay silent.
Right.
It changes how the genes work, but differently depending on the different tissues. It depends on what kind of tissues.
Yeah.
Genes that are affected in muscles aren't, of course, the same as those in the liver or in the duodenum, based on the paper, because they talk about three organs. Yes, the liver, duodenum, and the muscles, and compare it.
Yeah. So here, for example, in muscle, the ROH hits genes tied to stem cells and cancer pathways. In the duodenum, it targets hormone and transport-related genes. So here, basically, ROH can mess with some of the very important systems in the body.
And it doesn't stop nearby.
Yeah, that's right.
The ROH often affects genes far away. What scientists call trans effect. So trans effects, I mean.
Yeah.
So across tissues, more than 2,400 genes were influenced, and including those linked to the immunity, the development, and of course the reproduction.
Yeah,
In this
that's
part.
right.
Okay.
On the panel G, the coolest part, most of these ROH gene links are new. Bago lang. So traditional tools missed them, but the ROH analysis in this study uncovered hidden gene regulators.
That's why it's important, sir.
Yeah,
So
that's right.
that's why it's matter, because the ROH isn't just a marker of the inbreeding. It's a tool to discover new genetic switches and explain inbreeding depression at a very small level, or
Yeah.
at a very molecular,
Molecular
level.
level.
Mhm.
So from the whole pig down to the individual genes, ROH really connects the dots.
Exactly.
Yeah.
So to wrap up the studies here that we talk about, the effects of, or the runs-of-homozygosity on pig domestication and breeding by Tao et al. It demonstrated how the population history of pigs has elevated the homozygous regions carried in the ROH, influencing the economic traits, the gene expression,
Yeah.
and of course the fitness.
That's right. So here, the ROH analysis reveals how domestication and inbreeding have reshaped the pig genome, impacting those traits that you have mentioned, the gene expression, and even the fitness of the pigs. These insights are vital for improving breeding strategies, managing genetic health,
Uh-huh.
and understanding inbreeding depression at both the trait and molecular levels.
I agree with that, sir, and that's right.
Yeah.
It is important because it provides comprehensive genetic and functional framework for understanding how inbreeding and artificial selection have shaped the pig genome and what means for agriculture, to the animal health and biomedical research.
Yeah, that's right. So in broader impact for breeders first, this study offers tools to monitor and manage inbreeding, improving genetic health and economic traits for pigs. For scientists, it provides a model for studying complex traits, gene regulation, and even disease genetics in large mammals, not just in pigs. And for biomedical research, positions pigs as valuable models for understanding human genetic disorders and recessive disease. Because the organs of pigs is somewhat like similar to of humans. Yeah.
So it's a good model also.
Yeah, a basis for studying
For
further
studying.
research, especially in biomedical research.
Exactly, and it's a good model for us to understand how domestication works,
Yeah.
and how inbreeding were used to select the different types of traits that they want in the pigs.
Yeah, that's
Like
right.
for example, resistance to the diseases.
Yeah, that's some of the advantages of inbreeding. The breeders can choose what type of traits they want to be seen in their pigs. But then we need also to understand the negative impacts of inbreeding, like what we had just discussed, sir.
Okay, let's have the interactive questions for our listeners. So
Yeah, there are some
We
interesting
can have-
questions prepared here.
So, would you eat pork from a pig with 20 generations of inbreeding?
Especially those people who love bacon or whatsoever.
Exactly. And do you think the ROH analysis should be used in human medicine?
Yeah, that's a good question too.
It's a good question. So, what's your favorite weird genetic trait in animals? Do you think if you were a pig, would you rather be wild or
That's a funny
domesticated?
question. Sir Bon,
Okay.
answer that question.
Exactly. Okay. Thanks again for tuning into Science Side Up. If you've enjoyed this episode, I want you to share it with your fellow science nerds, farmers, or anyone who loves a bacon and biology.
Yeah, and remember, the genome is a storybook. ROH just help us understand or read between the lines.
And if you got questions or ideas for the future episodes, we're going to have one.
Yeah.
I want you to drop a message or tag us on our social media.
Yeah.
Social media ba?
Yeah, you can follow me at IG Jerwin.
And of course, until next time, stay curious and
Maybe don't marry your cousins.